thank Asim for inviting me to speak here. And I tried to put together a short talk so that there would be time for discussion. And I was hoping that every single person who came to the meeting would stay till the end, but obviously that's not happening. So my lab works on type 2 diabetes. And I have to beg indulgence from the UW people, because I always use the same introductory slides. So you've seen the same introductory slides to introduce type 2 diabetes. So I think you all are well aware, if you hang out at enough airports, that we're in the midst of an obesity epidemic. And with obesity, almost invariably, people become insulin resistant. And when they do, they have an increased requirement for insulin. And in the majority of cases, they meet that requirement by producing more insulin. And there are a lot of ways that that can happen, which I'll talk about a little bit. And if they do, then they manage to achieve a situation where they are not diabetic. They have normal glucose levels, but they still are insulin resistant. And that's described as the metabolic syndrome, and it has accompanying it a lot of problems: hypertension, hyperlipidemia, and even though they're not frank diabetic. But in about a quarter of those people, insulin production does fail to keep up, and that's when you lapse from this condition that we call prediabetes to frank diabetes. And it's a huge world epidemic. And there's a lot of genetics involved, and my lab is really interested in the genetics behind this process, which I like to summarize with this really simple slide that says what I just told you. Most people who are obese are insulin resistant, but they're not diabetic. Only a small number of them become diabetic, even though most people who are type 2 diabetic are obese. And there are a lot of potential mechanisms involved. Here's a short and incomplete list that includes insulin signaling defects, problems with beta cell proliferation and survival, the unfolded protein response, reactive oxygen species, vascular function, and we've actually found a gene that affects vascular function that's involved in diabetes, cytokines, adipokines, very exciting recent work on microbial communities in the gut, innate immunity. But the reason for doing genetics is because we can't imagine the complexity of all of this, these processes, and we really do need the serendipity of genetics to help us find our way. And what we do in my lab is we use as a model system 2 mouse strains that replicate this obesity-diabetes dichotomy, namely the black 6 mouse, which when made obese is non-diabetic, and the other strain is called BTBR, which when made obese is very diabetic. And this is summarized here. You're looking at blood glucose over an 8-week period in females and in males. And you can see there's a very drastic difference in the 2 strains' responses to obesity. So we can study this onset of diabetes in this very short period of time and look at changes in gene expression. We've done quite a lot of that. And we've also done a lot of genetics. So we take 2 approaches to this problem. One is positional cloning, where we make F2s, we map genes, we make congenic strains, we get down to intervals, and in those intervals we identify genes. And I'm not going to talk about that today because it's not as clever as a lot of the things that you heard in this meeting, but it does work, and we've found some very interesting genes with that approach. Another approach, which doesn't take quite as much time and actually requires some newer methods of mining data and making inferences from the relationship between changes in gene expression in various genes, is what we call gene expression networks. And so I'm going to tell you about 2 types of experiments we've done, what I call a time dimension experiment and then what I call a genetic dimension experiment. In either case, these are contrast conditions that challenge genes or gene expression changes to either be coordinated with one another or not. And so from the structure of the correlation patterns that we see in gene expression, we can identify and tabulate what we call gene expression modules. So for example, here's a summary of our time series experiment. There are 3 variables. Body mass, the mice were either lean or obese. Strain, the mice were either non-diabetic, black 6, or diabetic, BTBR. And age, 4 week, when the mice that will become diabetic are pre-diabetic, so any changes that we see anticipate diabetes and therefore are not consequences of diabetes. And 10 weeks, when one of the 8 groups, namely the BTBR obese mouse, is diabetic. And that's shown here. This is the only group, there are 8 groups here, 2 strains, 2 ages, lean or obese. The obese 10-week-old BTBR mouse is diabetic, has very high glucose because although they did increase their insulin, they didn't increase it enough. This is how much you had to increase it to prevent diabetes. And they have fewer islets. So we profiled 6 tissues in each of the 8 groups, and we grouped the genes in what we call modules, because in the 8 groups, but if you look at the color-coded modules here, they have the same red-green pattern of gene expression. There are 15 different ways of seeing differential expression when you have 8 variables, or 8 groups. And so we put together 105 of these gene expression modules across these 6 tissues. And here they are again, showing a subset of the ways in which you can have differential expression. And now we've separated the 2 strains, and you can see that some of them remain and are well retained, and some of them change as a function of strain, as a function of whether or not you're destined to become diabetic or not. But what got us most excited was 1 module out of the 105, which is shown right here, where in the non-diabetic animal, it was sensitive to obesity and age, but in the diabetic animal, it was sensitive only to age, and it lost its sensitivity to obesity. In islets, in adipose tissue, it retained its sensitivity to both obesity and age. Very much the same genes, but very different regulatory pattern in the different tissue. And so so this is the expression pattern, and this is the first principal component of that pattern, showing that it's sensitive to obesity. In the B6 strain, the non-diabetic, goes down with age, but loses that sensitivity to obesity in the diabetic mice. And here's an actual measure of DNA proliferation. The animals drank deuterated water for 2 weeks, and then we looked at the enrichment of deuterium in DNA. This is with Mark Hellerstein at Berkeley. And you see that, in fact, the gene expression changes do predict the actual proliferation that's measurable. And the same happened in adipose tissue. They both retained their sensitivity to obesity and age. Both strains did in adipose, as predicted by the gene expression changes. So we created what we call a gene expression network model of diabetes. These are the partial correlations between the modules within tissues and between tissues. And we put this on an interactive website where you can click and find all the genes in each module and look at all the expression, the correlation coefficients. You can also type in your favorite gene and see its expression pattern across all the 8 groups. So it's become a very useful model for people in this field, website that is. The problem with these schemes is that they're non-directed. So these are lines, they're not arrows. They don't tell you anything about causality. And we're very interested in causality. And that's where genetics comes in. So we did an experiment, took us 4 years to do, where we made 500 F2 mice derived from these 2 different parental strains. And then we profiled 6 tissues from those mice. And one way to think about what happens here is that in the F1 generation, meiosis is like a slot machine where you're randomizing the genome and generating an F2 with recombinations that try to maximize the genetic variance. And the greater the genetic variance in any phenotype, the greater your chance of detecting heritability. So for these three clinical phenotypes, this is a histogram that illustrates the variance. And what we want is very large variance, and we did get it. We saw a huge variance in glucose. This is a log scale for insulin, so it's 2 logs of insulin and a very large variance in number of islets. But most importantly, we profiled all the genes interrogated on a microarray in each of the 500 mice in each of the 6 tissues. So it was about 3,000 microarrays. A lot of data. And the largest number of mRNAs that were correlated with insulin, not surprisingly, were in the islets. That's where insulin is produced. But interestingly, in adipose tissue, and that turns out to be very exciting because adipose tissue we now know and didn't know 10 years ago is an important endocrine organ. And so it also implies that there's communication between adipose and islets. And I won't go into that, but we're very interested in trying to use our methods to understand inter-tissue communication. So what we do now with the DNA, with the RNA data, is we genetically map. So traditional genetics is, in the simplest term, just multiple regressions of phenotype on genotype. And wherever there's a correlation between phenotype and genotype, you know there's a nearby gene that might be controlling that phenotype. So now what we do is flip our definition of phenotype to be mRNA abundance. So now we're mapping the genes that control mRNA abundance. And they come in 2 varieties, cis-acting and trans-acting. If they map to the physical location of the gene itself, we call that cis-acting, and a simple way to think about it would be a polymorphism or a SNP in a promoter. Or trans-acting, where we're mapping to a region that's separate from the physical location of the gene. And it gets very, very interesting when there are multiple mRNAs that might map to the same location, because that starts to suggest a pathway or a master regulator with many targets of that master regulator. But by doing this, and this is what we see here, a common master regulator and multiple targets. Now, I can summarize a tremendous amount of data on one slide, which shows you in a 30,000-foot vantage point the genetic architecture of 4 tissues. And what I'm showing you here is on the x-axis is the physical location of, the y-axis is the physical location of every gene, and the x-axis is the location of the controller of that gene. So here, y-axis is where it lives, x-axis is where it's controlled. If those 2 are the same, you get this diagonal, and those are the cis-acting genes. They're controlled where the gene is located. But where you see these vertical stripes, these are what we call the trans hotspots. This is where a single locus can control the expression of many genes. And the z-axis, the intensity of the color, is the strength of the evidence, the LOD score. And what's really fascinating, and we're trying very, very hard to understand this at a statistical and genetic level, is why there are such really phenomenal hotspots. This one here on chromosome 6 in the islet is the hottest of our hotspots. And there are quite a large number of mRNAs whose expression is controlled by this one hotspot. And it does not exist in any of the other tissues. So it's not an artifact like a SNP in probes or any strange thing like that. It's tissue specific. And I stay up at night thinking about it all the time. Now, going back to the one module that I showed you earlier, which is the one that controls the proliferation of beta cells, which is really important in determining whether or not a mouse is going to develop diabetes, we found that that also was heritable. And here it is now, not as a function of strain, but as a function of genetics, because across the F2, it cohered. There was correlation across the F2. And now, we can categorize them as we can subdivide them or bin them as the ones that had the lowest expression or the highest expression across the F2, and ask, what does that really matter clinically? And the answer is it does very much, because the average glucose, insulin, number of islets, C-peptide, triglyceride, all these clinical parameters were highly significantly different between the mice that had the lowest expression and the mice that had the highest expression of the cell cycle module. But the genetic question is do these map to any part of the genome? And the answer is yes. In islets, they map primarily to 3 locations on chromosomes 2, 4, and the hottest of the hotspots on chromosome 17. And so we're really focusing a lot of our work now to try to figure out what is it on chromosome 17 that's coordinately regulating the expression of cell cycle genes in the islet. It's really interesting that about 75% of these transcripts in the islet overlap with the cell cycle transcripts in adipose tissue, but the genetic architecture is completely different. And so here you can see that, because the genomic hotspots that are controlling very much the same genes in adipose tissue are on different chromosomes. In this case, 1, 6, and 10. 3 different chromosomes. So now the last thing I'm going to tell you about is what we do with networks. So if we take a subgroup of those transcripts, and here I'm only showing you 11. Here are 11 transcripts that are controlled by the locus on chromosome 17 in the islet, and they're totally dichotomous with regard to their response to obesity. So all of the ones, all of them are increased in response to obesity in the B6 strain, and the others are either decreased or not changed at all in response to obesity in the BTBR strain. So they're dichotomous in their response to obesity as a function of mouse strain. So let me tell you how we do networks, causal networks, because we want arrows and we want causality from our data. The challenge is, if you have 2 phenotypes, in this case mRNA abundance, that are highly correlated with each other, what you want to know is phenotype B changing as a consequence of a change in phenotype A? Is phenotype A changing as a consequence of phenotype B? Or is there an independent model where they're not really changing in response to each other, but they're changing in response to a common factor that's influencing both of them? So those are the 3 models that we try to evaluate. And because we have genetics, we can actually resolve these 3 models in the following way. We take advantage of one very simple principle, which is that the relationship between genetic polymorphism and phenotypic changes is a one-way line of causality. So that gives us an anchor point. Oh, it's already time up. I thought I was giving a short talk. 3 more minutes. So the question then we can ask is if we want is the direction of this arrow. And we take advantage of the fact that we have a locus controlling A and we have a locus controlling B. And these come in 3 flavors or 3 genotypes, where the genotype affects the gene that's being controlled. In other words, that's how we define heritability, that genotype affects phenotype. So now we can do a causality test where we look at the effect of a change in genotype on B through A and the change in genotype on A through B as a function of the genotype at each of these 2 loci. And that can be scaled up, but to a limited extent, to simultaneously solve all of these equations for all of these genes. It's extremely intense in terms of computation. These computations take days to compute. And this is all the work of a statistics graduate student working with Brian Yandel named Elias Chaibub. And so then he computes a likelihood ratio for this relationship, which tells us what our confidence is in the direction of those arrows. So here's our friend again, the cell cycle network, and here's a network that he computed showing you the loci that are regulating the various transcripts, and then the relationship, the causal relationship. Each one of these has an arrow between the transcripts. And I just want to say that we're focusing on one gene now, on chromosome 17, that happens to be a nuclear membrane, an integral nuclear membrane protein that looks like a strong candidate as a master regulator. And so finally, I obviously didn't have time to tell you what else we're doing with networks, but we have heritability data on microRNAs. We're going to be generating some heritability data on proteins. We already have some from mass spec analysis. We've done metabolites by mass spec. And of course, we have lots of clinical traits. And so we're trying to now overlay these causal networks. So we're not only looking at causality between mRNAs, but causality between various categories of phenotypes across tissues. So this work is a team effort. We benefited from the resources of Rosetta being led by Eric Schott until recently. Rosetta is actually now defunct. But this allowed us to do such large-scale experiments that we wouldn't have been able to afford with our own funding. So we're grateful to Merck for that. And then I've been working very closely for more than a decade now with Brian Yandel and Christina Kanjorski, two statisticians, and their graduate students. And then Mark Keller in my lab is a leader on this project, and we've been joined in the last couple of years by Carl Broman, and very recently by his wife and his brother. So it's become a Broman family affair. And I'll stop there.